A very good day to lecturer and fellow friends. Today our group is going to present the journal about distribution pattern and phylogeny of marine Stramenopiles in the North Pacific Ocean. The abstract of the journal is marine heterophyte nanoflagellate are small unicellular eukaryotes typically ranging from 2 to 20 micrometer. They are thought to be important grazers of bacteria and phytoplankton and to contribute to nutrient cycling. The aim of the journal is we investigate mass growth in 2 North Pacific provinces. We qualified mass 1A, 1B, 1C, and 4 cells with specific fluorescent in situ hybridization probes and explored their relationship with environmental parameters and other microbes in data from 2 ECS cruises.
Now let's look at the introduction. Marine Stramenopiles are a diverse suite of eukaryotic microbes found in marine environments. Several mass lineages are thought to contain heterotrophic nanoflagellates. However, mass remains uncultured and data on distribution and trophic modes are limited. Among the mass groups, mass 1, 3, 4, and 7 have been found in both open ocean and coastal systems on the basis of 18S RNA gene clone libraries. Relatively few studies have investigated the abundance and 18S RNA gene diversity of eukaryotes and more specifically mass in the North Pacific Ocean.
The methodology are study site and general sampling, nucleic acid and microscopic sampling, nutrient and CHL measurement, flow cytometrics analysis for phytoplankton abundance, microscopy, pre-observation of mass 4 by tertiary addition, DNA extraction and clone library, sequence and phylogenetic analysis.
Let's look at the results. Characteristic of study sites. Mass diversity was investigated at oceanic provinces on either side of the MPSG. The Eastern China Sea lies over the Asian continental shelf, while the CALC region is off Central California but extends northwards to Alaska and south to Baja California. The latter includes the eastern boundary current of the MPSG and a highly productive inshore upwelling region. In each of these provinces, 3 sites were used to represent different habitats for mass populations. Specifically, we sampled inshore at highly productive sites Eastern China Sea and MBARI time series, with the latter being a mid-bay site in the Mountain Bay. The major stations in the 2 provinces had distinct characteristics and were subject to different seasonal influences. Surface water temperature in the East China Sea ranged from 15.8 to 25.3 degrees Celsius in spring and 23.3 to 29.6 degrees Celsius in summer 2009, while salinity ranged from 27.9 to 34.7 and 23.8 to 34.1 in spring and summer, respectively.
The second result is distribution of mass 1 and 4 cells in ECS. Very few mass 1A cells were detected in the ECS station and none were detected in the plump area, with the exception of a single station in spring. In contrast, mass 1C cells were observed frequently at greater abundance. A significantly positive relationship between the abundance of mass 1B and 4 was observed.
The third result is mass cell size, biomass, and prey. Cell sizes of mass 1 and 4 from the smallest to largest was mass 4, mass 1b, mass 1c, and finally mass 1a. Mass 1a cells in the ECS were smaller than in the data from high latitudes, where the cell size was 7.7 plus minus 0.3 micrometer, but our values are based on very few cells.
Next, in the part discussion, mass 1b and 1c as well as mass 4 had peak abundance in the plump area during summer. Therefore, we compared mass abundance between what we define herein, using salinity measurements as coastal water and more oceanic waters using correlation and t-tests. Mass 1c abundance was higher in the coastal water than at the more oceanic sites, and its abundance was negatively correlated with the nitrate and phosphate in the oceanic samples. It appears that mass 1c and 4 thrive in the inner shelf of the ECS, and this may be related to higher prey availability, higher productivity in general, or other environmental parameters associated with this site.
In conclusion, our results indicate that many of the previous observations on the distribution and phylogency of mass lineages hold true in the North Pacific Ocean. Between the Stramenopiles, many trophic modes exist, and this is presumably the case across mass as well. Their ecology, physiology, and diversity are still little understood, in part due to their uncultured status. We found that the cell size of mass 1B and 4 vary with temperature, indicating that different ecotypes may exist within their lineages.
The second conclusion that we get from the journal is finally mass 4 organisms have been shown to consume heterophyte bacteria. Here we observe the consumption of phytoplankton, especially Syntricoccus. Combined with this statistically significant relationship observed between the natural abundance of mass 4 and Syntricoccus, this may indicate that the latter serves as a food resources for mass 4. The factors that shape prey selection by this mass lineage and now mass 4 influence their ecosystem roles, particularly with respect to control of primary producer communities. That's all from our group presentation. Thank you.